So I can go ahead. Okay, so first of all, I want to thank everybody that made it to this meeting this evening. I can see some dignitaries here, but sorry, I cannot introduce all of you one by one. I actually want to give special thanks to my supervisor, my present supervisor, who also promised to be present here. I don't know if she is here already, Professor Luisa Berai. She is a wonderful supervisor, a very supportive supervisor. I don't think she's here yet, but I know she might join later. So I will take this time to also sympathize with those of you who do not have wonderful supervisors, or rather accept my condolences, because it's wonderful to have a supervisor that is very supportive. And so I also thank all of you who joined from all parts of the world. So we can start off. The main reason why I wanted to hold this section, can I share my screen now?
Yes, you can. You're the host.
Can you see my screen?
Yes, please, click on present. Click on present.
Oh, okay. Sorry, I'm not used to modern facilities. So tonight we'll be discussing molecular docking, and I'm actually surprised that okay, everything is scattered actually. This is the outline. So we are going to introduce the concept of molecular docking. We are going to discuss how to formulate a research question. Please, just give me 1 second. Sometimes you can mute those in the background that their microphone is on. You can mute them yourself.
All right, let me do that quickly.
So please, the reason why the group was created is so that you can direct your questions there. It would not be very fair to message me directly why the session is still on, because I'm getting lots of messages from people asking questions directly in my chat. I may not respond to them anymore, so I can concentrate. So we talk about how to formulate your research questions, and we talk about how to identify your targets and how to acquire them from the appropriate repositories. Then we go to the docking proper, which is actually the main reason why we are here. We do the preparation of the proteins and the ligands. Then we do the blind docking. Some other time, as we will agree later, we'll do another one called the site targeted docking. It's different from blind docking. And then we also go to the post-docking analysis. We do the minimizations, the result interpretation, the 2D and 3D presentations. We do everything tonight. I hope that we can be able to do this in a few hours so we can all go to sleep. So to start with, what is molecular docking? So according to this reference, Dar and Mel, molecular docking is a kind of bioinformatic or computational modeling, which involves the interaction of 2 or more molecules, that's the target, to give the stable adult. What it's saying here is that molecular docking is a bioinformatic tool or a computational tool. It's not a laboratory assay. It is kind of simulation using the computer to predict the binding of two molecules. These two molecules could be two proteins. It could be a protein.
Hi, Darlington.
Hello?
I'm sorry, we can't see your screen.
Thank you very much for calling my attention to that. So, I think I did the wrong thing, so I'm gonna share my entire screen. Can you see my screen now?
Yes, we can.
Thank you very much for calling my attention to that. So, molecular docking is a bioinformatic tool, like I said, or a computational tool. This means that it is based on computer simulations. Computer simulations to predict how two molecules bind or interact with each other. It could be two proteins, it could be gene products, it could be a protein and a ligand. It could be someone asked me if you can still do protein to DNA binding. It's possible. But for tonight, we are going to limit our discussions to protein and ligand binding. When we talk about protein, we are talking about, it could be a receptor, it could be an enzyme, it could be any other kind of protein, and then it interacts with a ligand. A ligand could be a molecule, a drug, or whatever it is. So like I already stated, before you attend this training, you should know a little about protein ligand interactions. So this is our receptor ligand interaction. This is, we are trying to predict how these proteins or receptors interact with ligands. Then we want to predict the, what is it called? I think I'm going ahead of my slide. So the aim is to determine the binding affinity and binding conformation. So we want to determine the affinity of the ligands to the protein, and we want to see the conformation in which it binds. There could be different conformations. In fact, some ligands, some models use the lock and key model. Some others use the induced fit, meaning there's some flexibility, and it's very good to use induced fit model when we do binding, so that we can actually see different conformations that these molecules, these ligands can bind to the protein. So in the binding affinity, I just need to state this, that the more negative binding affinity, the more likely it is to bind. That means in binding, we are desiring to get a highly negative binding. So a binding of -5, for instance, is better than a binding of -3. The binding should not be plus. If it is plus, it means there is no binding. Bioinformatics, or rather molecular docking, is useful in several different fields, and that is not the aim of this discussion tonight. You can read up those ones at your own time. It can be used for those who are in pharmacology. It can be used in lead optimization studies. And when we talk about lead optimization, when you find a drug, the parent drug that you find is called the lead compound or the lead drug. From that lead drug, there could be some modifications to the lead compound to get better drugs. That can, it could be by adding some groups or by removing some groups from the lead. So you keep modifying the drug until you get the one that would not present side effects and would actually do exactly what it is required to do. So it could also be used in pharmacophore modeling, which is like when you have a scaffold of a drug, and then you want to model it. How do I explain this? You model the scaffold, to get some other activities from it. It could be used for such studies. It could be used in many studies, but these are not our aim tonight. Our aim is to find out the interaction, both the conformation and the binding affinity of a ligand to a protein. I want to say that what we are going to do, or molecular docking, does not in any way replace laboratory assays. Just like you have in vitro tests and you have in vivo tests. Even if you find an activity in vitro or in vitro, you still need to do the in vivo analysis to know if this actually holds, if it will actually work in the living system. It cannot replace it. The same way, when you're doing molecular docking, you are trying to screen possibly hundreds or thousands of molecules to find which ones can give you the activity that you want. But when you find it, you cannot say that we have gotten a drug. You must go through the normal laboratory assay. So it's more like a preliminary study where you screen so many compounds and then you pick the best ones which you can now take to the laboratory. It saves you time, it saves you money, it saves the environment. Because if you run, for instance, if you're doing in vivo study on thousands of compounds, you are using more animals to do these studies, which is not, human to this animal. There are so many laws coming up now about using animals for tests. In fact, some scientists are of the opinion that we should restrict tests to stem cells and not the use of animals. This is very good so that you save the use of animals in these tests and use only a few when you have found the main ones that you want to use. It also saves chemicals. Imagine you are running in vitro activities or tests on several compounds, the amount of chemicals you use. But here you can screen all of them just using your computer and then select just a few. So there are so many advantages and there are some disadvantages as well. One of the disadvantages is that this is computational, of course. Sometimes the computation might be wrong. It might not give you exactly what you want. It might give you false negatives at some times. So so whatever you do here, you must for sure subject it to laboratory assay before you can be able to say that yes, this is what we have. Okay. I want to explain exactly what I think somebody is annotating the screen. Today, the aim of this discussion, because I'm going to talk about how to formulate a research question. The aim of this discussion is because I understand that so many of us in Nigeria, we have this desire to go into science. We want someone to hire us in his lab to do research or even as a master's student. But there's this saying that people only invest in talents, or I don't know how they say it. You must have to prove yourself first before someone can hire you and invest in you. So some of us don't have that opportunity to go into the lab and do some experiments to show, or studies to show that we're actually good in research. I know that some of you have already left school and you cannot go back to the lab. So I usually encourage people to do review. You don't need to go in the lab. You can do, collaborate with people, do a very thorough review and publish it. It can show that you are very good in research, okay? So I also thought of this. This is a way that you can even do a research without going to the lab, to the laboratory. So you can, now we are talking about how to formulate a question. You can think about a disease. Let me use a typical example, the COVID-19. You can think about the COVID-19 and think about how can we produce a drug for COVID-19? Fine and good. Now, first of all, because we are talking about how to get our targets, first of all, you need to understand the mechanism of action of the disease you are into. Let's say COVID-19, let's say measles, hypertension, whichever one. You need to understand how does this disease work. So you need to actually read, you need to actually study published literature on the field and find out what is the mechanism. How does this pathogenesis of the disease, is it a disease that is caused by a pathogen? Is it a metabolic syndrome? You need to understand how this works. If it's a disease that already has some medications that are working on them, you need to understand what are the mechanism of action of these medications, what are the drawbacks, and how can we improve these drawbacks? So these are kind of questions you are going to look into. I will take COVID-19 as well and continue on it. Let's assume that for COVID-19, you decide to study what are molecules that can be targeted to COVID-19 as a drug to improve the management of COVID-19. Then you start asking yourself, how does COVID-19 actually work? I'm saying this because I already have read some things about it. It's something you can research on your own. We know that it is a pathogen, a virus that causes it, the SARS-CoV-2 virus. So because it's a pathogen, the best thing to target as a drug to this disease is something in the pathogen, not in the human being. because not all proteins in the body can be targeted with drugs. Here, let me start by explaining what is a protein or target or enzyme and its druggability. When we say druggability, is ability of a gene or gene product, such as a protein, to be targeted by a drug. And not all proteins can be targeted by a drug. As you can see in this chart here, more than about 74% of proteins in the human body cannot be targeted by a drug. They are not non-druggable. In fact, they are not even disease-related in the first place. They have no relationship with disease, so they have nothing to do with drugs. But about 16% of these proteins are disease-related but still not druggable. It could be that they are not druggable because we are talking about the human proteins. It could be because these proteins are, let's say for instance, enzymes that play a key role in the body of the human being. So you cannot target it with a drug, because if you target it with a drug, it means that the person will also be affected. You might actually be stopping some physiological roles in the human being while you are trying to target to cure a disease. Well, maybe I'm being too blunt in saying you cannot target them with a drug. What I should have said is this: if your aim is to find a drug that will cure the disease without affecting the individual significantly, then you shouldn't target these proteins. If on the other hand, your aim is, I want to cure this disease, even if it means that this person will even die, I will even kill the patient before I cure the disease, no problem, then we are free. You can target these undruggable proteins. But there are approved targets. These ones are the ones that already in the market, they are being targeted, and there are drugs that have been targeted on. These are human proteins, not even the pathogens proteins. These are human proteins. About 672 of them are already targets that have drugs in the market. Then there are about 7% that are potential targets, but no drugs have been, they are just potential. You can target these ones. You can even target the already approved targets. It depends on what you're targeting them at to make a better drug. Okay. So, like I was saying, if it's for COVID-19, you may not even need to start targeting any, because if you, let's do something here. Let's go online. Maybe I shouldn't do this because of time. What I wanted to do is to go online and search the mechanism of infection of SARS-CoV-2 virus. You will see that SARS-CoV-2 virus infects the human being by attaching its spike protein to ACE2. ACE2 is angiotensin converting enzyme 2. Angiotensin converting enzyme 2 is a very key protein in human metabolism. You cannot inhibit that protein. If we inhibit that protein, COVID-19 cannot enter the human body. But if you inhibit the protein, you might also kill the person. So we don't target it, okay? Then the best thing to look at is a protein in the virus itself. And every medication for SARS-CoV-2 has been targeted at the spike protein, because that is what it used to gain entry into the human body. So it's targeted at the spike protein of it. That's how you start off. So you now see that, okay, now what I need to target, or what has been targeted, which I can also follow, is the SARS-CoV-2. Then you can go online and find out what are some ethnomedicines that has been used to cure COVID-19. You can find the list of them. Maybe in Nigeria, you can find up to 40 of them. And then you also try to find out what are the bioactive compounds in these plants that could have elicited those responses. Then you find a list of them. You can now try to dock these compounds to the protein and see its binding affinity to the protein. From there, you can find out, or you can make a conclusion that so and so compounds might be good targets for developing COVID-19 drugs. You have found a very good research that you can work on. So this is generally, you can find a better way to find a research, but this is a way you can start to think of a research problem that you want to work on. And tonight we are going to work on, what is it called? Inflammation. I don't know how to remove this, and it's very annoying. So,
okay, so if you click on annotation on your screen, then you click on clear all annotations, it will disappear. The delete button on under annotations.
No, it's not even the annotation I'm talking about. It's this okay, there should be a way to hide it. Don't worry, you can go ahead with it. So now we want to go to the problem we are going to talk about tonight is inflammation. We are going to see how does inflammation work in humans. Good night. So we type Can you see my screen?
Yes, you can.
Okay, I don't even have time to read all of this. When you are working, you need to do a proper research. I'm just trying to do something very quickly. I go to maybe the images. Let me look for another image. So all of them are giving mechanisms of inflammation, but okay. This this is already even showing me that this is how inflammation takes place. You have membrane phospholipids. It's acted by phospholipase A2 to give arachidonic acid and cause, this is cyclooxygenase, can act on arachidonic acid to give PGG2, prostaglandin, all these prostaglandins and thrombosins and all of them. And these are the molecules that lead to inflammation. And you can inhibit cause with this drug, resveratrol, and that can inhibit inflammation. From this, when you read properly, this is just, I just did something very quickly. When you read properly, you can find out that this cause cyclooxygenase is an enzyme that is really implicated in inflammation. And by targeting this enzyme, you can, in fact, some drugs for treating inflammation are actually inhibitors of this cause. If you still go deeper, you might find out that there is cause 1 and cause 2, and that one of them is cause 1 has some other functions that are important, while cause 2 is what you really need to. If you can selectively inhibit cause 2, it's even better than inhibiting all the cause, because if you inhibit all the cause, then you have some side effects. Okay, but we are not going into that. We are just trying to identify that cause is an enzyme that has been targeted by some drugs that are used to cure inflammation. Now, your own might be, what can we do? Can we find out what are some of the locally used medications in Nigeria, these herbal medicines that they use to cure inflammation in Nigeria? You may want to find out that. For you to find out, you can, first of all, you might say mechanism of action of, sorry, this part we'll do it quickly so we can go into the main meat of discussion. Mechanism of action of anti-inflammatory drugs. Okay, let me see. So here, like I said, all of these are targeted at cyclooxygenase to prevent conversion of arachidonic acid to prostanoids. The same here, these are non-steroidal anti-inflammatory drugs. It's targeted both at cause 1 and cause 2 to prevent conversion of arachidonic acid to prostaglandins and all of them. From what I already said, you can see it here that this this cause 2 is actually what leads to inflammation and pain, fever. Like, this is actually what you needed to have inhibited, not really this, because if you go further, you find out the effects that this is going to have. Okay, we've passed that stage. The next thing we want to ask ourselves is, what are some ethnomedicines that have been used? First of all, please let me go back so that I don't mix these things up. We were talking about how to identify the proper targets for our proper protein targets. And we have seen that if we want to target inflammation, one of the proteins we can target is the cyclooxygenase. That's what we have confirmed from here. Then the next thing is we want to think about what are some targets that we can use against it in this our study. So the targets of the ligands or the drugs have a quality called drug likeness. That's ability of the ligand to act as a drug. You cannot just pick a ligand, any ligand, and try docking it against a protein. There are some things that you must confirm. Even the protein itself, there are some things that makes a protein to be what's the right word? To be druggable. We call it druggable target. And I'm going to read out some of the things you should check about the protein before you start targeting it with a drug. First of all, the docking of the ligand to the protein, of course, should be feasible. It should be able to dock, it should be able to bind to it. Another thing is that binding should lead to a physiological response. It should lead to some kind of modification in the protein. And that modification in the protein should lead to an improvement in the disease condition that you are into. In this, what we are doing here in the first place, you might be able to find out if this ligand can bind to the protein very well. For you to find out the physiological response is going to bring and the effect is going to give might require further studies. Okay, for what we are going to do tonight is to find out the binding affinity. Then the target itself, we talk about drug likeness. It should be drug-like. That is, it should have the ability to act like a drug. You should have the ability to target to the protein that you want to target, and you understand, and affect the same things we have talked about, this physiological response and giving the activity that we desire to give. These are things that should stand out. You might not know, for instance, you might still know, because when you find a ligand, you can read about it online to see what are the properties of this ligand, and then we are going to do that. We've already seen that we can identify the cause as our protein of target, because we know for sure that it has already been targeted by some drugs, and there are some side effects, like we have already said, of targeting, because some of these drugs are not selective between cos1 and cos2. So these are things you can know. You can think about any disease you want to work on, and then think about how to, some things about it. So for us to pick our target this time, we can now ask ourselves, what are some local medicines that people have been using to cure inflammation? Because sometimes some of these local medicines that they use, it might be effective because there are some bioactive compounds in it that are eliciting the response that you want. But just that the local and traditional users, the ethnomedicine practitioners or whatever they are, do not really even know what they are doing. They are giving bioactive compounds, but they do not really understand what they are giving. And you might find out what they are by doing some literature review. So we can search for okay. Here, remember, like I said, you need to do a proper study. I am not really here to do the proper study now. I just want to pick something. Here we can see this one says species. It's not very specific. This one says, which includes, okay, let's look at this. I saw some plants there, but I already have decided the one we are going to use. I mean, if you can't find it here, we can use any other one. to do the experiments tonight, but so for you who is going to do a study, like I said, you should be able to take the time, really investigate what has been done in that field so that you don't just, it's not something like what I'm doing now to just open online and look at one or two works. No, you really need to take the time to really investigate and see what has already been done. Okay, so here we have this species of this and this, but they did not give any.
Hello, sir.
Yeah?
Hello, sir.
Hello?
Hello, can you hear me, sir?
I can hear you. I can hear you. If you're referring to me, yes.
Hello. Hello.
Can any other person hear
I
me?
used a plant. Okay, I used a plant. I used a plant in my undergraduate.
Okay.
So
Hello? Hello?
I think you can continue. His network is hanging.
Okay. So we see this plan. It says research reports shows that leaf extracts of this was effective in treating these and these and these isolated compounds from these and inhibited prostaglandin synthesis, which is exactly what you want to, achieve. You want to inhibit prostaglandin synthesis. So let's see. We copy this and we ask what are the bioactive compounds in this. So it's already here. Is reviewed such as alkaloids, be specific. Okay, I will just open any of them. So here is still saying tannins, alkaloids. These are not, I want the specific compounds. So I cannot see specific compounds here, and I don't have the time to read the entire work. So we move out. Have I checked this? Yeah, check. Have I checked this? Let me see. Okay. Tannins, and it's still groups of compounds and not specific compounds. Then we modify our search and we say anti-inflammatory compounds. So it's gonna be actually a search. Like you need to really calm down and look at what has been published on this, so you can find out actually what are the compounds that are present in the plants that you have found, This is if you want to work with, if this is what you want to research on, of course. If you want to do any other thing, you can, but this is just like give an idea of how you can find out a research problem that you can work on. I can't see any compounds here, so I am moving out immediately. Let's save time. Let's just save time. We've already spent already spent 47 minutes. No, we can't continue. So in my search earlier, I was able to find some compounds, and I think I'm going to just pick one of them and we continue. When you have the time, you can, take the time, search for the plants and the compounds that are present in them, and then you can know exactly what compounds you can make a list of them. You can make a list of up to 40 compounds from different plants that are reported to have anti-inflammatory effect potentials. If you're not working on inflammation, if you're working on diabetes, anti-diabetic potentials, you're working on COVID-19, whichever thing you're working on, you understand, it's just an example. I'm using inflammatory. So you can find these plants and then you can go ahead to download the protein of your interest and the targets of interest. The plant I found, the compound I found in the plant I saw earlier is called, in fact, I even doubt I know because I just did that this afternoon. I even doubt I can remember the name of the compound. So somebody was trying to speak up before, and I was hoping that he could tell us the name of the compound so we can use it for. Okay, I think I should find it.
Okay, I think he posted on the chat room that we should look for Calancho pinatium. It actually inhibits inflammation. So you probably could check that out.
That's his, okay. Does he know the compounds in the plant? Let's start with the plant first. Can you pronounce again, please?
Calancho pinatium. K-A-L-A-N-C-H-O-E. K.
Even if I am a robot, I wouldn't capture what you just.
K-A-L-A.
Okay.
C-H-O-E. Then pinatium.
Okay.
P-I-N-A-N-N-A-T-U-M. Yes.
It is called Calancho pinatum, but yes, before it was known as Biophyllum pinatum.
Please, can you spell one? Can you spell the last part of it?
P-I-N-N-A-T-U-M.
P-I-N-N-A-T-U-M.
Yes, that's it. That's the first one under Google search. Yes.
So okay. Yeah, and the name of the compound that inhibits inflammation, it is called, I will type it now, but it is mouthful. It is called stigmast something. But I have the journal.
Just spell it.
No, let me type it. Okay, I should spell it. S-T-I. S-T-I.
Good night. T-I-S-T-I. Hello?
No, S-T-I, not T-I-S-T-I.
S-T-I-G-M-A-S-T.
S-T-I.
G-M-A-S-T.
This.
Stigmast.
Please, James, can you spell it for me? I don't get it.
All right. So S-T-I-G-M-A-S-T. Yes. Continue, Fabumi.
-4.
That's it.
-4.
That's it.
Okay.
That's it. 4 and 3 1, right?
Yes.
That's it.
Okay,
I'm
fine.
trying to.
That is, it's okay. Now, we've identified a protein.
I've dropped it on the chat. I've dropped it on the chat.
It's okay. You have already given us the name of the compound. That's all we needed.
Okay.
So we found that the protein we are going to use is the cos, and we found that the compound we are going to dock for tonight is this compound. Fine and good. We go back to our work. The next thing is how to download these targets. How do we get them? There are a list of repositories that you can get your proteins and your targets, or even to create your own proteins and targets if they are not available in 3D formats. For the proteins, you can use the Protein Data Bank, which is PDB. It's like, I think, the highest. It has a lot of proteins there and even some nucleic acids as well. You have the PubMed, which is controlled by NCBI, National Center for Bioinformatics Information, Biotechnology Information rather. You have the UniProt, you have the ChemBL, you have the Swiss model. Swiss model is a place where you can even model your own proteins. Okay, there are so many of them. I just put just a few because this training is not about that. It's just to give a head on, and then we go to the docking. Then if you want to get your ligands, you can go to drug bank. You can get so many, all the drugs actually are there, and some other molecules, some other ligands are there. Good night. You have PubChem, which is the same, almost the same with PubMed, the same NCBI. You have ChemSpider, you have ChemBL, the same ChemBL here. You can get proteins, you can get ligands. You have NPAS, and so many of them. Okay, so you can use these to download. If you check in one, you don't see, you can check in the other one, you might see it. So for instance, if you want to use, to get our protein, we can use Protein Data Bank. When you go to Protein Data Bank, you need to type in the name of the protein you want. We want to get the cos and we want to get the human cos. You can use another model if the human version is not, it has not been done or it's not online. You can use another, but I mean, you cannot be very sure that they have exactly the same activity, although they are the same enzyme. They might be, of course, they should be, what do you say, enzyme catalyzes the same activity, but might have some differences between them. So let's just check if we have the human cyclooxygenase. Okay, so here we have human COS1 crystal structure, which is good. And I'm going to show you, you can go down, you can see so many of them, so many of the same, they're all the same thing. It's just that sometimes they are crystallized with some other compounds. It could be some other molecules that were crystallized alongside them. It could be that they are crystallized in different chains and all those things. So you need to understand what you are downloading. For instance, this is a crystal structure, of course. I can click on it, and these are the things you need to take note of once you get a protein. You need to check. I can see that it's Homo sapien. I can see that it's an oxidoreductase, which is what cos is. I should look at the resolution. This is very important. The resolution of which means the resolution, we know what is, I don't know how else to explain it, is the resolution of crystallization. This is 3.36, which is very, very high. I cannot say that this is the rule as to how high the resolution should be before you accept it. Some people say up to 1.5 is good, 1.8, 2.0, 3, but of course 3 point is very, very high. So for me, I think it's more or less, you can use any one, but the higher, the better. I think that's all I can say about the resolution. But you need to know it because you must report the resolution of what you are using. You come down here and you check. This is, you can know more about, this is the DOI of this in PubMed, but the DOI of the people that's the publication where they crystallized this, because we'll see more of this when we do the site-directed docking, because you need to go and read how this compound was crystallized before you can do the site-directed docking. So you need to can go here and read more about the publication. These are the people that crystallized it and did the work. You need to check which one is the chain of the enzyme you are using. This is prostaglandin GH synthase, which is the same as COS. It's a prostaglandin synthase. That's the actual name of this enzyme, and it is our chain A. So you should take note of it. This is the sequence. We don't really need all the chain length and all those. So you need to know the resolution. You need to know the chain. It's chain A we're interested in. If we go down, we still see that there is a chain B, there's a chain C and D, and there are some other ligands that are crystallized with it, just like the fluoro LC, what is it? Fluorocitrate anion was crystallized with it. We don't need all this, so we are going to remove them when we start docking. So we have seen the resolution and we have seen that what we are interested in is just the chain A. You can download this protein here, come here and download PDB, protein data bank format, PDB format. You can click here and it has downloaded. Okay. Another thing you need to take note of, very important, is the ID of what you are downloading. This is the PDB ID. Is 6Y3C. So I need to write it. I do not have my pen. So please, somebody can help me type it somewhere because we need to refer to it. 6Y3C. So we've downloaded it, and this is how it is. Oh, it's going to open with Discovery Studio. No, that's not what I wanted to do. I wanted to open it with Notes. So this is it. Truth is that I don't understand this laptop, so let's just continue.
No, click on it first, then you see open with. Click on it, let it be highlighted. No, it's not highlighted.
If I click on it, it will open.
No, now click, right click.
It will, okay. Because I know, oh, sorry, my normal computer, I set it that I just click once and it opens, so I cannot.
Good, open with. Go down, down.
Okay, yeah. Okay, I forgot that.
No. Click on look for apps.
I know.
Down,
so you can take a look at the protein. It's very long. You can see here the chain A, which we do in the next edition. All these are the chain A. If you want to even edit it, if you want to prepare this protein manually here, you can prepare it by taking only the chain A and removing from chain B. Here you see the termination is where the chain A ends. You can remove all others and also remove everything before the chain A. Okay, we learned that some other time. For now, we just leave it this way and we go back. Then the next thing we need to do is to download. Can you still see my screen?
Yes, we can. Site 2, section 2, right?
Man. The thing is that I don't. I was sharing. Please, I'll stop sharing so I can know what I'm doing. Okay. Yeah. So I'll share again. So we've gotten our protein. We downloaded the PDB format. Don't forget. If we downloaded we went to download and we downloaded the PDB format for our protein. Then we go for our ligand. We talked about, here is the presentation. We talked about We can use drug bank if you want. We can download from PubChem. We can go to PubChem. So I think I copied the name of that your drug. Oh, please, can you tell me the name of the drug again, of the compound?
Stigmast for something. Stigmast.
Stigmas 4-1-3-on.
Stigmast 4-1.
Stigmas 4-N-3-on. That's the second one on the list. It just disappeared now. Yes, that's it there.
Okay, so this is it. If there is no, this compound is not available, you can create this compound yourself using Compound Discoverer, using ChemSketch, or there are some other tools. Not Compound Discoverer, rather, Discovery Studio, and all of them. So this here, you can get some information about this compound. You can look up what the compound does, its IUPAC name. You can also look up its canonical smile. So this canonical smile is very, very important. You can download any compound using its canonical smiles. If you copy it, if you go anywhere and paste it, it's going to give you the compound. I don't know. If you go to PubChem, for instance, and paste the canonical smiles, it should give you the compound you are looking for. Did I copy it correctly? Yeah, I did. Is this a synonym for this thing we are looking for? Synonyms, it's not. I mean, I'm not going to defend PubChem because it's their business, it's not mine. I know that with canonical smiles, you should be able to get the compound you are looking for. Okay, with this, you need to download here. This PubChem cannot give you the PDB format of this, but it can give you. The problem is that the 3D format of this is not available. What we have here is the 2D format, and we don't need the 2D format. So we need to look for another compound. Do you have another compound? Because you cannot draw now. In fact, drawing is what I've never done because I've never had the need to draw a compound. Do you have another compound?
Okay, we can check. You know, when we are checking that
before then, wait. Okay, please.
That stigma actually has 3D. That stigma. Yes, it does. The stigma has 3D. Yes.
No, it's not this. It's not this. What's happening? Let's start again.
Yes.
Stigmast 4. Okay.
-4E3. The second one.
Oh, sorry. I'm getting old. So we save the SDF format. the 3D SDF format. We've gotten it down here. What I can do now is this. Oh, sorry, I did not check the ID. So the ID of this compound, you can help me note it down as well, is the PubChem ID is 5484202. 5484202. I think I need to get my pen now. Please, just a second. Okay, so five four eight four two zero two. Now we've gotten our, I can hear you.
Okay, please. The last one that I gave us, I really didn't get the name of that, the human whatever, but I got the ID, like the 6Y3C, but I didn't get the name of the thing. I don't know if I can just
It's cyclooxygenase. I didn't expect you to know the name of the protein I'm downloading. What I expected you to do is to know the name of the protein you want to use, because I want to start docking, then I allow you to think about a disease you care about, and then think about the protein of interest and download the protein and start docking your own along. You shouldn't download the one I'm downloading. It's better that, well, for now, I understand that maybe it's not very easy. So it's cyclooxygenase. Cyclooxygenase, just COX, COX. So you can just type human pox, COX, and get it. But later on, think about your own disease, not disease you're suffering from, sorry, the disease you're interested in, and the compounds through search, the compounds that have been targeted to this disease or that are present in and plants that have been used in curing this disease in the traditional way, you can think about them. Okay, so we've downloaded our molecule as well. Another thing I wanted to do is to go to drug bank and see if I can see this molecule, because here now we did not, of course, in PubChem, you cannot see PDB format. In drug bank, let me see if it's available. I am seeing this name. Let me see if it's a synonym of this. It's called cytostenone. It's called beta cytostenone. It's called for that's taking me to where it's taking me to where I don't know. So I think it's not the same as the one I saw in drug bank. I don't think it's the same. So possibly this is not a drug. So we did not see it in drug bank. Okay. So if it's the standard drug, for instance, for cause, for inflammation, some of the standard drugs are aspirin is a standard drug. So if you come here, because one thing you can do in a study to make a study standard is that you can compare the binding affinity of these compounds to the ones of the standard drug that is used for disease and see if the compounds will even give you a better binding than the standard drug. So we can search for aspirin, for instance, as our standard drug for this. And aspirin is here. It's already told you that it's a salicylate. used to treat pain, fever, inflammation, migraines, and all of them, and all of these. And if you want to get it, this is the Ascension number. You can also get it down. The reason why I'm writing these numbers is that we are not really going to use the ones I'm downloading. We see a way to just get it directly with the number when we want to talk. But if you want to download it here, you can go here to download 3D, and then you can download it in PDB format. You can download this. So you can see it open. In Drug Bank, it's going to open this way. It's not going to download the file for you. All you needed to do is to Control A and Control C. Then you go to your notes, your notepad, and you paste it. And when you paste it, you name it as aspirin.pdb. Okay, we've gotten aspirin. So let me see what we have gotten so far. In my downloads, we have the aspirin. We have, this is the compound that we're interested in. And the name of this compound is what's the name again?
Stigmas 4, 3, and 1.
Stigmas, I think. So let me see if I do .pdb. It did not change it to a PDB file. That's the problem of using this as my default app. I'm going to change it. Well, let's continue. Now we've gotten our proteins. We've gotten our protein, we've gotten our ligand. Then the next thing we need to do is to go into the docking experiment proper. What I have done so far is that I've introduced what molecular docking is all about, and I've tried to show us how we can think about starting a research question and to raise a research problem, and how we can get our targets and how we can download these targets in appropriate format from repositories. We can download our protein in PDB format, very important, 3D PDB format. Our ligand we can download in PDB format, or we can download in SDF format, always 3D. And then when we get the SDF format, we can change it to PDB format by renaming it. It's just unfortunate that what I will do is I will download it with my other laptop and then copy it and bring it here, because this one has already set Discovery Studio as the default app, and it does not allow me to do what I want to do, and it's very annoying because this laptop I'm not used to it. So I'll download here. Okay. So now we are done with the first session of this training. We are going into the practical aspect. If you have any question while I download this, please ask your questions, and I am going to address them quickly. Once I finish downloading this, I don't need to download it. I remember, I don't need to download it. You can go work directly with it. So ask your questions before we start the next section. Please, does anybody have a question?
Please, I have a question, sir.
You don't need to call me, sir. Just say Darlington.
Oh, Darlington.
Ask your question. Yeah.
Okay. I wanted to ask, downloading from PubMed and Drug Bank, do you need any software, or you just go to Google and put PubMed and then go search the drug and just download for you to download in the PDB format?
You just need to go to Google and go to the website of PubMed or whichever one you want and download. It's just to get a file from those repositories. You don't need any software at all to do that.
So once you save it in any file, you save it as the PDB format already?
Sorry, I did not get the question.
Like after downloading, you get to save like a document. You go and the way you did the file for it. Once you're saving it, it's already in the PDB format.
When you download it, you're already downloading a PDB. Like, let me repeat. When you go to PubMed, for instance, when you see download, you see the 3D, you see download, you see several options. You can download the mode, you can download SDF, you can download 3D SDF, you can download the PDB, you can download the conical smiles, which I talked about before. We clicked on PDB. So what is going to download for us? Of course, PubMed does not download, it only opens it. Then when you copied it, when I copied it, I saved it as .pdb. So it's saved as a PDB. If I downloaded from this site, for instance, I go here and I download as 3D SDF, it's going to save download as 3D SDF. If I download from, what was the other site? We downloaded from no, the first one we downloaded was from what?
PubChem.
Data PDB. It's going to, PDB was the first one. PDB, you have options of what to download as. You can download as FASTA file, which is just a plain text. You can download as PDB format. When you click on PDB format, it downloads it as PDB already for you. You don't need to change anything. If you cannot download as PDB and you can download as, but I mean for your protein, you should download as PDB. If for your ligand, you cannot download as PDB, then you can download as SDF and then change it to PDB by renaming it as .pdb. I don't know if that is clear. If you need to copy it from where it is opened as a test, you copy it, then you go to your notes and you paste it and you save that file as name.pdb. It's already in PDB format.
All right, clear. Thank you.
Thank you very much. Please, just a second. I need to close this door because I am. Okay. Please, any other question before we continue? Any other question? No?
Hello, good evening.
Good evening. Please go ahead.
Please, what if the compound is not on PDB? How are we going to do it?
Sorry, please, can I get the question clearly?
If the compound is not present online, the compound found in the plant we are using.
Yeah, if it's not present online, but you know the structure of the compound, you already know the two-dimensional structure of the compound, then you can create the compound. There are so many tools that you can use to create the compound yourself, the 3D formula, the 3D, the 3D, what's the right way? The 3D file of the compound. You can use Discovery Studio. You can use, there are so many other tools like, I think it's ChemSketch or something of that nature. There are so many tools that you can use to do that. But I've never done that before because I've never had the need to do it.
All right, thank you.
If the compound, the structure of the compound is not known, then you cannot do anything about it. You cannot use it. For instance, in my present thesis, I did LCMS and I found so many compounds which I know the molecular formula, but not the molecular structure. There's nothing I can do about it. I mean, you need, so definitely you can't do anything about it. You can't use it. You need to use the ones that the structure is known, but if the 3D format is not available, you can create it. You can even upload it when you create it for others to use it subsequently. I don't know if that is clear.
Yes, thank you.
Thank you very
Hello,
much.
sir.
So,
Hello, sir.
yeah, I can hear you.
Sorry, sir. Please, I'm trying to download this stigma something from the PubChem now. So, as in, it was only in SDF format. There is no PDB format there. So, can I convert the SDF format to PDB format?
Please just watch the screen. Can you see my screen?
Yes, I can still see your screen.
This is the compound
Okay.
you're talking about.
Yes, sir.
You go to download, you go to 3D, you save this 3D format, SDF. Now,
Okay.
if you have not already made, I need to change these settings. I just don't want to do it now. Like I said, I just got this computer today and I downloaded this software. So I've not set what is my default app for what. If not, if it was in my other computer, and I can do it in my other computer so you can see it, James, can you make me admin with the other system so I can do something with it now?
Okay, so you're actually the host, right? And you can make your other account the host, yes. Just go to the meeting participants, go to Zoom, participants, then click on the other one and then make host.
Thank you.
You're welcome. All right, so you have to remove the volume of the other
Yeah,
line.
but I think it's okay now. Is it?
Yes, it's okay, very okay.
Okay, so here. So this compound is. Oh, I'm not sharing my screen, sorry. Can you see my screen?
Yes, sir.
So this is the compound I just downloaded now. All I needed to do is to go here.
Okay.
And rename as stigmas dot pdb. It has gone to a PDB file.
Okay. Just by renaming.
Yeah, just by renaming it.
Wonderful.
Oh, it's still
Great.
so sorry. No problem. I can open this file. I remember. What you can do, if you want to convert to PDB, you can open it, then do Control A, Control C, do Control N, that's new file, Control V, then you save it as tig.pdb. Okay, so this now is a PDB file. So you could copy the contents of the file itself, open it with your notes, because if you open it with Discovery Studio, it's going to open the 3D for you. Just open it with notes, copy the contents, create a new file, paste it, and name it as .pdb. The reason why I didn't know this is that I didn't really need to do that, because I can start preparing it as SDF file without really needing to convert it to .pdb file. So that's it. I believe that this is clear.
Yes.
Thank you very much. So I'll go back to the so okay. Now what we are going to do now is to go to the docking proper. And we are running out of time already. This is already 22:30, which is, I think, 10:30. So to start the docking, we need to open, let me share my screen. We need to open some software. The first thing you do before you start any docking studies, and it's good that this is a new system, is to go to your documents or wherever you want to and create a folder that you will do everything you want to do. You create a new folder for whatever you want to do in your docking. You create this folder and whatever you are doing. So if you wanted to take these downloads, you needed to copy them and take them to this new folder and place them inside here so that everything you are doing will be inside that folder. It's very, very important because you might not remember to navigate between these folders, and if you don't get them to that folder, at some point your docking will terminate. It will not run to the completion. And the first software we are going to use in our docking experiment this evening is our Chimera. So just open your Chimera if you already have it installed. I am thinking of how can you be watching me and also working on your system. If there's a way you can divide your screen, fine and good. If there's no way you can divide your screen, you can follow along because we are already running out of time. You can follow along and later you can do yours. So this is my
She's trying to say something.
I didn't hear you. I didn't get you. Can somebody hear her clearly, please?
whatever
I said that when I install the camera, it doesn't need to pay for license. I can't use it.
Please, you can type your comments and someone can read it
Just
out.
type your request. Yeah.
So when the first time you open the camera, you see a black screen like this, then you can go here to file. So what we first do, if you can see, I will send you the PDF I prepared, the PowerPoint I was using. So the first thing you need to do is we are now in docking, okay? The first thing you need to do in docking is to prepare your protein, and then you prepare your ligand, and then you dock your protein and your ligand, and then you do post-docking analysis. After the post-docking analysis, before you now start trying to do presentation and interpretation and all of that. You don't need to pay for any license to.
Darlington, please excuse me. Tosin and Fabumi, please mute yourself. We are getting feedbacks from you guys.
Oh.
Sorry, go ahead.
So the first thing you should do is to open file, and then you already link it to the. You go through to get to the folder that you have created for it. You understand? Then you select the protein from here, but I am not selecting the protein from here. Okay, what I would rather do is to fetch the protein by ID. So when I've gone online and I've seen the name of the protein I want to use, I didn't really need to download the protein or the ligand. I know that the protein, I wanted to get it from Protein Data Bank, PDB. Please, James, can you tell me the ID of the protein we wanted to use? I didn't write it down. I wrote from the ligand. Sorry?
6Y3C.
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 6Y3C. So I fetch. That's the protein of our interest. Had it been you downloaded the protein, this is exactly what you would get. So how do you prepare the protein? It's very simple. You go to select and you go to chains. Remember that we saw online that this protein has chain A, B, C, D, and we said we are only interested in chain A, which is the cyclooxygenase. So we select chain A, and now we need to delete the other chains B, C, D, and all of them. So we go back to select and we do invert all models. So it's more like reverse the selection. We selected every other thing apart from chain A, and then we go to actions, we go to atoms and bonds, and we delete. The other one is gone. You can say, what of these ones that are here is not part of the chain? Well, apart from the protein, remember that apart from the chains, remember that there are also other residues, other ligands that we are docked with this protein. So we go to select, we go to residues. You can see all non-standard residues, which includes the FLC that we saw, which includes the water molecules. You don't delete any standard amino acid, which is part of the protein. So you can say all non-standard, and it has marked all of this. And then you also go to action, atoms and bonds, delete. So what we did is we selected the chain. It's only chain A remaining now because I've deleted the other ones. We selected the 3 chains that we do not want, and we deleted in actions. But how we did it is that we selected A, and then we reversed all selections by doing invert all models, and then we delete all the others. We also did the same for the residues. We selected all non-standard residues apart from the standard amino acids, and we deleted in the actions. So what we have here is only our chain A and nothing else. Okay, you can turn it around to see it and all those. Then you now go to, you can view it in several ways. Okay, you can go to presets. You can see it as ribbon. You want to see different sections of it. It colors it to see different parts of this protein. You can view it as atoms, which might interest you, so that when you are preparing it, you will see the changes that are happening to it. You can view it as hydrophobicity, the surface, this is also interesting in reporting because, for instance, if the active site is here, you can see how your molecule will fit into the active site, because the active site is like a holo in the protein. This might be a way you want to present when you finish docking. We talk about presentation of results, okay? So you can also have publication forms. We talk about those ones when we get there. But let's keep it as atoms for now. And then we go to tools. You go to surface editing. Let me tell you something you do. Please do this so that you don't forget it when it is time. Before you start any docking studies, remember I already told you that you need to download a folder called Vina. It contains 3 files. And I also said that you should cut that folder and paste it in your program files, or wherever you installed Chimera. You should paste that folder of, it's in the instructions for the installations. So you should have done that already. For instance, this is my C. This is my local file C. This is my program files. This is where I installed my Chimera, and this is also where I pasted my Vina. You must have done this before now. If you have not done it, then you need to download Vina. It's a folder. It's there in the instructions for installation instructions. You download it and you paste it here. So let's go back. So when you start Autodoc, the first thing, when you start this Chimera, the first thing you should have done is to go to tools, go to surface binding analysis, go to Autodoc Vina, then executable location, you pick local. If it's not in local, you go to browse, and then you find where that Vina is and pick Vina. It's in the wrong place now. My Vina is in, I think it's in users, HP, not documents, but
downloads.
No, I don't think it's in the my Vina. It's not the files I downloaded, it's the Vina itself. It's in my program files.
Application data.
Application data. No. Oh. Tools. Something binding analysis. Autodoc Vina. Yeah, it's actually in the right place. So why is it HP documents? For you, it wouldn't be as difficult as it is for me, for obvious reasons. So just set it to your program files.
So your program files is in the first documents, in the first folders, not HP users.
Oh, yeah. Sorry, Vina. So you now pick this vina.exe. It should be only 3 files there. Forget that this was when I was trying to test run stuff that I saved things here. It should be 3 files. Pick vina.exe and save and close. So it's in vina.exe. So you have done this, then close this. Then let's go back to what we are doing with our protein. So you go to tools, you go to surface editing. Here you are going to add hydrogen, add charges, do this. But instead of doing these things one by one, you can do everything by going to minimize structure. Now it's asking you, this is the protein that we downloaded, because I did not rename it. Remember, it's directly from the internet. It's not the one I downloaded earlier that I renamed. This one is directly from, and the name is 6Y3C, which is the name of the number. You need to report all of this in your work, because this is like the level of refining you are going to do to this protein. So if you set it at 100, which is the default, I don't know if the default is 200, you can check. It gets refined, but the higher you go, the better. So if you set it at 500, for instance, it gets much more refined. And if you set it at 500 and set this at 50, but remember that it will take time. This step might take 1 hour. I don't know how, for the system I was using, it takes at least 1 hour. Even if I put it at 100, if I put it at 100, 500, it might take 3 hours to complete just this step. Okay, that is why so for the test, for the sake of this demonstration, let's just put it at 10 and put this one at 1. But I mean, this is not something you want to do in a standard work. So you must report this, and this, because whatever you use will affect the results you will get. The interval you don't need to report is just like telling us after how many steps should it tell us, should it give us updates that this has been done and all those. Fix atoms, none. Memorize choosing options for subsequent times. You can memorize, it's nothing bad. For the others, no, then you minimize. Then this is the same protein we have chosen. It says, what should you choose for the following? You want to delete solvent? Yes. For me, it doesn't really make much sense to delete solvent because I know that we are trying to mimic the biological system, and in the biological system, there is always aqueous, and whatever is happening happens in the presence of water. But the explanations are that if you don't delete solvent, well, if you don't delete solvent, you will not have actually the interaction that you want. So you need to delete solvents to see how things actually are. Well, I accept. So we need to delete solvents, and we need to delete all non-complex ions. And if alternate locations, set all of them to change. These are non-standard to standard forms of these. Incomplete side chains replaced with this. I don't really understand what these are, but from all the training I saw, you can just leave this. I mean, changing it to other things does not really make so much difference, so you can leave it. You want to add all hydrogens, and you want to add all charges. We don't need a Mo2 file, so you can leave it. And then you go, okay. Then it says, consider each model in isolation from others. Not really important here because we are only doing one model. Later on, we are going to do 2 models. And then that time we need to untick it so that they will be saved as one file. Okay, but for now, we can still untick it. It doesn't matter because we have only one model. So metals stericonly, consider also hydrogen bonds is yes, which will make your system slower, but no problem. Residue, name base, this is not something I don't really understand till now, but it's always, I mean, always left this way and leave these ones, and you click OK. The last step, it says charges. These are different ways of calculating these bonds because this is computational, either this or this. I've tried both of them and it makes no difference actually. Maybe it makes differences in some things, but not in all the works I've done. And non-standard residue, you can leave everything as it is. You can pick this gas Teiger if you want. It doesn't make much difference. And okay, remember you report every step you have here. So it's going to tell you that your system is not responding. It might tell you that your system is not responding. But down here, you can see after each 10 steps, because that's what I choose, it will tell you that 10 steps have been completed, 10 steps have been completed, and all those until it completes. Even if you say your system is not responding, don't close the system, leave it, it will respond. It might take 1 hour, 2 hours, 3 hours to finish this step. With this system I got now, I think it will take us less than 10 minutes. I mean, especially as I put it at 10, it should take us less than that to complete. It shouldn't take time. But this is not what you want to do in your work.
While we are waiting, can I ask a question?
Yeah, sure.
Aside from camera, what other software can we use for this process? Because it's like mine is telling me I need license, so I'm missing out.
Yours is telling you need what?
License.
I think you downloaded the wrong app. See, sorry, the app, if you're not in the group, the link I gave to camera is a wrong link. I later sent the right link. And if you're using Windows, I already sent the real app in a drive. You needed to download that one. The link you saw in that installation guide is wrong. It's not the camera that you need.
Okay, thank you.
Please, Darlington, I don't know if it's possible they can be sent to the group because I joined the group this evening and I didn't see any of the messages. I wasn't in the group earlier. Sorry about that. I didn't have
I'll
the
send
link.
it right now.
Thank you so much. Thank you.
All right, so Mr. Darling, thank you so much for how far you've gone with us. I was wondering if you could just take a recap of the preparation of this protein again. I think I got lost from where you talked about the VINA being in the Chimera tools and everything you did within that period. I didn't really follow because I was trying to put the VINA folder in the Chimera folder and program file. So I was wondering if you could just take that again as we still wait for this. Thank you.
Please, I'll respond to you. Just 1 second, please. Thank you. I've sent the link for the camera, so you can use this to download the app. Okay, sir. So you said you got lost at what point, please?
Okay, I said I think I followed up to where you removed the non-essential bonds and all those stuff. But what the things you did after you explained the need to put the VINA folder in the program file, I think I didn't really follow up. So I can't really tell what you did because I had not done that previously. I didn't get that information. So I just had to put that folder there now. So the only thing I was able to follow was when we removed the non-essential, I think, atoms and bonds, the non-essential chain, and then the non-essential residue. That's where I followed up to.
It's okay. So why
just to add to that, sorry, can you just do a quick recap of everything? Is that possible?
Okay. Yeah, I can. It's possible. So I would.
Yeah,
what I will do is I will use the second system to do what I have done before because I can't do anything on this system now. It cannot do anything. If it's even saying not responding, so nothing will work. I will use the second system so you can see everything I've done once again.
Yeah, that would be perfect because I've actually not followed up on whatsoever you've done so far because of this issue of network.
Yeah, but this system, this session is recorded, and the record will still be made available so you can follow along sometime later.
Okay, that's perfect.
So I am sharing this other screen. So I'm going to open so what I did, okay, I thought it was me, it was the computer. Okay, so when you have worked on some compounds, the compounds are going to be listed. You are no longer going to have a black screen. You are going to have compounds listed here because, yeah, that's the things you have worked on will be listed here. So you go to File, you go to Fetch by ID, you go to Fetch by ID, and then use PDB and say CY3C. If I don't know if CY3C. Is that correct?
Yes, it's correct.
Fetch. Sorry, let me start all over again. Let me close this section. First of all, once you get your camera open, the first thing you do is you go to Tools, you go to Surface Binding Analysis, you go to Autodoc Vina, you go to Executable Location, you set it to local. It can be here. When you get it, you set it to local, and you go to Browse, and you find where your program file is. For me, it's in the users, Jojo, documents. No, it should be Jojo. Should it be documents? No.
Downloads.
No, it cannot be downloaded. It should be downloads. Oh, it's program file. It's program file in the very first place. It's program file. This is where I installed my camera, and that's where I pasted my Vina. So you don't need to go inside here. Where you go into this one downloads is when you want to look at the files that you are going to work with. It's a different thing. The files you are going to work with, I said you need to create a folder where you put all your files and all your works. It's a different thing. The Vina is where you installed Camera, because this Vina is a plugin to Camera. So wherever you installed Camera, you also go and paste your Vina there, and it's in the program files. So that's where your Vina is. So you need to go there, open Vina, and click on this .exe. So let me double click. So this has plugged in this Vina inside your camera. You can say, okay, you can just close if you want. So that's like the first thing you need to have done. So now you have prepared your plugin. The Vina is a plugin in the well. Then you can now go to files. I can just open this directly because no need to start downloading again. What I did is to select, and I looked at the chains, and I wanted to, you can start deleting manually, go to B, say actions, atom/bonds, delete, you go again, you select C, you do the same, but a simple way is to select only A. Another way you can select, look at a way you can select this, hold down your control and click on any part of the molecule you want to select. Then, still holding control, click the up key. It has selected just one part of the molecule. Then you click again, it selects the entire molecule. So this way, I have selected all my chain A. I did not even select the residues. Remember that right now, if you go to residues, we see all these non-complex ions are there. But as I've selected just the chain A, and I click on invert all models, and I go to actions, atom/bonds, delete. I've deleted, I don't need to go to residues again once because I've deleted all those non-standard residues. I've deleted all the other chains as well. Everything has been deleted. It's only chain A left here. You might say, why are we seeing some of these bonds? Some of these bonds are like they're important bonds that the people that crystallize this wanted to show. It's not as if it's not part of the chain A. It's not another compound under to chain A. It's only chain A that we are seeing so far here. Okay, I am done with the protein preparation, so I'm going to go back to this other system. Okay, well, let me show you what I did. When I have done this, you can view it in any way you want. That's not important. You go to Tools, not Surface Binding Analysis this time, but Structure Editing, and you go to Minimize Structure. You set everything here. You can keep it wherever you want. I just did it very small so it can run fast. And you need to report this for 1, 2, 3, 4. You don't need to report this. This is just like how much update you want to get after. You can set this to this if you want, then you minimize. So you get the video and then, or when you run your own, you see these things very much more clear. This system is very slow. That's one thing I hate about it. It's even here. Then it gives you options. I want to delete solvents. Yes, I want to delete all non-complex ions. If alternate locations keep only highest occupancy, I select everything. Incomplete side chains, I select it. I select add hydrogen, I add charges. I don't need to write a Mo2 file, so I don't select it. I click OK. Consider each model in isolation from others. I have only one model, so I don't even need to, whether I leave it on or off, it doesn't make any difference. I want to also consider hydrogen bonds. And this, I clicked it. I did not take this. I don't know if it makes any difference. Okay, it likely doesn't make any difference. I was not expecting. Oh, yeah. So it has added hydrogen bonds. If you look at this now, if you look at this, you will see that hydrogen bonds have been added to it. Because some of these things were not here earlier. You may notice it. Let me see when we go to chances, when we go to residues. So you see that the structure actually changed. So the last step would have been to click OK here, but I don't want to do that. Once you click it, it starts running, and that means I've condemned this system for today because it's going to last more than 1 hour before it finishes. So I say close. Let me go to this other system and continue from where we stopped. I don't know if this makes it any clearer now.
Yes, it does. Thank you.
Welcome. How do I? I don't. This is confusing. Okay. So I'll share my screen once again. Now, the docking is done. What we need to do is to serve this protein as PDB. So we go to File, we save PDB. Then I'm going to save it inside this folder, like I said earlier. I'm going to save it here as the name of this protein is cos. No, this is prepared cos, so that I don't mess it up. The reason is that if I had downloaded the course earlier, I must have saved it as course, and I can mistake both of them. So this one is the prepared course.pdb. Yeah, you should always click this add surface.pdb. So with it, I don't even need to add this .pdb, but yeah, I mean, you can never be wrong by adding another .pdb to it. Self-displayed save. You don't need all those, just save. Now I have saved my protein as a prepared protein. I need to do exactly what I've done for this protein for the ligand. The ligand wouldn't take time. It's a very simple molecule. So we saw this ligand in PubChem as SDF file, and the number was 5484202. I can fetch it. Oh, and I did not even close my protein before downloading my ligand. Let me close this section. So here is my ligand. If you checked PubChem when we did this thing, you would see that this is also the same. This is that ligand. Look at the structure. It has, this is it, the ball and stick model. And here it is. You can see it's the same thing. This is the round chain. Let me call it round chain because I don't know if all of us here are chemists. That's it here. Now these groups, you can also see them here. So for sure, this is that compound. So we do the same thing. We don't need to select chain because it has no chains. It's not a protein. You don't need to select residue. It has no residue. It's not a protein. All you need to do is to go directly to tools, structure, editing, minimize structure. I will use exactly the same settings I used for my protein, for my ligand. You will see this ligand, how it changes when I prepare it. Everything is, because I said, remember my settings. So everything is still remembered, everything. Okay. So this would not take so much time. And it's going to change the shape, just as it did to the protein, it's going to do the same to the ligand. The protein, maybe we didn't notice because it was so tiny and we didn't see the changes that happened to it. While it is running, if you have questions, you can ask so we can address it. So what we are doing, can you hear me? Can somebody hear me?
Yes, we can.
Okay, so what we are doing in this preparation is that we are trying to make this protein to be in the best form to be able to interact with a ligand. And what some of the things we do is to add the hydrogen bonds, because we need to add these polar groups, the protons, the polar groups, and all those things, so that the groups in the ligand can bind to them. Binding is done by, for instance, a serine group binding to an OH group and all those things. So we need to have provided those things in the molecule to enable proper binding. The same thing we do to the ligand, and also give the ligand to be in the best possible conformation to be able to bind to a protein. So these are the things we needed to do. That's why we are preparing the protein and the ligand to be, to enable them to actually dock well when we do the docking. That's the essence of the preparation. So after we have prepared the both of them, remember that for each of them you prepare, you are going to save it as a PDB file. Even if you got it in any other file, like our ligand, we got it in SDF file, but we are going to save both in PDB file, and then we are going to dock the both of them, and the docking takes time. I think that it's completed. But I'm not very sure of that.
Hi. Once it finishes, is the protein supposed to change? I mean, for the first one you did.
It did change. Some things did change. Some groups were added, some groups were removed, some things were removed. It was changing as it was running. We didn't notice it. You could notice it if you were paying attention to it. You would be seeing the changes happening. But it's going to be the same protein. It's just that some tiny parts of it will be removed and some things will be added to it.
Okay.
Now you can see the molecule changed a little now, if you observed very well. And it's done.
Okay, thank you.
Thank you.
Okay, so a quick question, please. Why are we doing all these modifications and
Sorry, I cannot hear you because your network is very bad. You can type your question.
Why
Somebody
are
raised.
we doing that?
Sorry, hello?
Yes, I was the one who raised hand. Chinazadike.
What's the rationale
It's just
for
a
doing
little
that?
contribution.
What
In
is the
order to
modification
make
being
it
done?
visible enough for participants to see.
I said, why are we doing all the structure editing and then
I think two people are talking at the same time.
Guys.
Can you hear me better?
Yes, I can hear you. Can I go ahead to speak now?
Yeah, sure.
Okay, I said in order to make it visible enough for participants to see the changes that are occurring, the background could be changed from black to white. That's when it will be easier for participants to see the changes that have been occurring. Yes.
Yeah, but I mean, whether we see, well, sure, but we know for sure that we have prepared it and changes occurred, even if it's not something visible for us to see with our eyes. You understand? I mean, when I dock this, I start docking and I go to brew a cup of coffee and I come back and it's done. Even if I don't see the changes, for instance, I know exactly that something must have happened. I set the changes I wanted to take place before I left. Yeah, but thanks. Somebody else was asking a question, Israel. But you can type it because I think your network is bad. So we continue. So we serve our, please, just 1 second. I locked this door and this is a public place. I think some other people might need to. Yes, okay. Sorry. How are you? Hi. Hope you don't mind having. Hope you don't mind. So we serve as PDB as well, and I'm going to name this as my duct. I just name it ligand because that's ligand. The name is one kind. Duct ligand, sorry, prepared ligand instead. And it's still the same thing. And I serve. Now that I have prepared my protein and my ligand for docking, I need to upload the both of them to the same space and dock them. So my ligand is already here. All I need to do is to open, and this is the protein. So I have the both of them in the same space, the protein and the ligand. So I need to now see how they interact with each other. And I can do that by going to tools and go to surface binding analysis, which is the first step I was talking about, where you set this. You can still do it now if you want, but instead of going, yeah, you go to now Autodoc VNA. You need to select where this is going to happen, and it's supposed to be exactly the same folder, and you can name it as protein ligand dot, it should be a dot pdbqt file, pdbqt. The protein and the ligand, we saved each of them as pdb files. This should be a dot pdbqt file. I already ticked it here, and I set the location. Then I need to select which one is my protein. My protein is my prepared course, that's the receptor, while my ligand is this. Then the next thing is to, no, let me not use this so that we can be seeing what is happening here. So the next thing I need to do is to, now it is called blind docking because we need to define where is this ligand going to bind in this protein, which parts of the protein is it going to interact with. And we can use this resize search volume. Search volume is like, where it's going to bind using button 3. You can use any button, but if you use button 1 or button 3, it's gonna frustrate your life. And I'm telling you honestly, button 3 is what I have found to be effective in. So you can just put 10, 10, 10 for all of them because you are now going to adjust the volume until you get it right. So just start with 10, 10, 10 or whatever you want to start with. Okay. And you use 10, 10, 10, you'll see that a grid has appeared here. So you need to take this grid and fit everything here inside the grid. The grid shouldn't be too bigger than this, and all parts of it must fit into the grid. Like I said, button 3 is the best for me. I think I understand this better in a black background. So I bring it here. Now, this is where you must use a mouse, for sure. You cannot do it without a mouse because you use both the right click, the left click, and this wheel. I don't know what to call it. I think I can call it wheel. All of them have the function. Based on the button you are using, you can keep trying until you find out what each of them are doing. For instance, for this, I can zoom. How do I even shift this? Okay, yes. So I can zoom in this way using this, increase this. So this is gonna be a very tough part of the experiment to get everything to fit perfectly into the grid. You don't need to bother about this because it's not part of what we are. It's just the protein that should fit very well into the grid. I think I've done a good job already. So you must look from all possible angles to be sure that everything fits perfectly into this. Then you go and, very important, copy down the grid you are using. So what we ended up with is minus 34 point this. This is the X, Y, and Z direction, both the center and the size. So you must copy this down and you must report it in your work, because if someone wants to reproduce your work, he must use exactly the same grid that you used. So if you're working on 40 compounds, for instance, remember it's the same enzyme you're working with. So when you come to the second compound, you just need to copy these and paste them here and, run it. You don't need to start doing it again. You must make all of them to be exactly the same grid that you used in the first one. So this, for instance, is okay for me. I can use it. I think everything fits in perfectly. So I copy this and put down in a table. I set everything here through the smart changes. Now, okay, I think I'll get there. Advanced options, I set everything to maximum. The number of binding modes is how many, because you're going to get different binding affinities, like different conformations and their affinities. I want to take 10 binding modes and see the affinities. That's the maximum I can get. Okay, you might be interested in only the first one. Actually, in my result, I'm only interested in the first one, but I can see all 10 of them and see exhaustiveness of charge. You need to report this, of course, this you don't need because it's only one at the end of the day that you are going to use. There is, I think, a button I am skipping that says, okay, no, it's not here. It's not here. So I apply and then it starts docking. This is going to take a while. So at this point, you can brew a cup of coffee. It's already doing like my system is hanging. Oh, so if you have questions, you can ask.
Okay, like I was trying to ask before, I don't know if you can hear me clearly now.
I can hear you clearly now.
Yeah, I was saying that what's the rationale behind the structure editing? That's both for the ligand and the protein. And what is the implication if we don't do those editing, the minimization that we did? What's the implication if we don't? And why is it important that we do it?
Okay, so I think I explained earlier that this structure editing is a way to make the protein to be in the best possible, what's the right word? To make it to condition it to be able to really dock with the ligand. For instance, you need a ligand to dock to a protein using OH groups. You need to use serine groups. You need to use all those groups. And those groups have something they interact with. Without adding hydrogens to the protein and to the ligand, without adding all of those things that we added, the charges and all those, they might not interact. So you need to prepare them, give them the groups that they need to interact so that they can actually interact. You need to remove water because if you put water molecules, it might cause them not to interact. I already said earlier that this doesn't make sense to me when I was learning molecular docking because I told myself that I know that these things happen in the living system. I mean, in an environment that there is water. So I don't know why I should remove water while doing it, but that is how it is done. So this is also a reason why docking experiments cannot replace in vivo laboratory assays, because I mean, if you do this in the absence of water, how do we know for sure that when you in the living system it will be the same result? Because it will happen in the presence of water. But these are things you should do so that at least that you get the result that you, I mean, because without it, you might finish docking and it did not even bind anywhere. So you need to prepare really the protein and the ligand before you start docking it. I don't know if that makes some sense.
Yeah, it does. It does. The reason why I asked that question is because I know I've been seeing some works lately where people just do molecular docking, and sometimes I know these things are not easily reproducible, and as in the biological system. So that's why one reason I'm asking if we will keep removing all these and then to interact, whereas that's not actually the case. So sometimes somehow it looks as though why are we actually doing it? But then I get the point. It gives us an idea to know how to target our research. All right, thank you, sir.
Okay. Actually, if you're doing site-directed docking, it's a different thing. It's a different approach altogether. But for this, for instance, well, like I said earlier, we can never be sure that the computational method gives us 100% accuracy, because in the living system, some molecules will be protonated, some will not. Some will lose protonation along the way. So in the docking, we are doing it, we are protonating everything. So it gives us its own result. In the living system, it might happen that way, it might not happen that way. So we can't be exactly sure of how it happens. So this is the result is out. Let me close this. These are the 10 conformations from 1 to 10. The best one is the one with -7.3. The second one is -7.0. So you can see it till the last one. You can see how they interacted. I can move this to the side and play a movie. So you can go here, movie, play. So you can see each of these binding and where they were interacting in the protein, different binding conformations. So one other thing I thought about blind docking is that, for instance, one of these could be the best binding affinity. For instance, this could be the best binding affinity. But how am I even sure that this is happening in the active site of the enzyme? If it's not happening in the active site of the enzyme, then it makes no sense. It might still make some sense. I know that an inhibitor must not be competitive inhibitor, okay? It can bind in a different site in the enzyme and give it a conformation that it cannot bind the protein. So it can be a non-competitive or uncompetitive competitive. I've forgotten these things. I left biochemistry several years ago. So it makes some sense, but for me, I still like site-targeted docking.
The term is allosteric inhibition.
Yeah, allosteric inhibition, yeah. So I think it still makes some sense to target this molecule at the active site of the enzyme, which is what we do the next time, which is site-targeted docking. Because for this, what I would choose is just the one with the highest docking, the highest binding affinity, which is -7.3. Okay, and I have no clue if what I am done is the best thing to do. If this is the best conformation or not, I have no clue. I am only selecting the one with the best binding affinity. So for each of them, you can see what groups and all of those things. They are not really important for me. The most important thing is to save this one. So I can remove all others. I purge them. Then for this, I click on it and I go to File and I click Rewrite, not save as, but rewrite. And we're going to rewrite it as PDB-QT file. So I can name it my course ligand. Let me call it my doct course ligand dot PDB-QT. And I'm going to save. So that is just the best result. The ones we purged, you could save them as well because you might want to go back and see them. So I can unpurge them. It's possible to put no. I know a way to bring them back, but let me just leave it for now. You can list all. Is it? No? You can find that out at your own time. I tried that some time ago and I saw a way to bring them back after purging them. But that's not really important now. So what we have done is that we have prepared our protein and ligand, and we have docked the protein and the ligand together. The next thing we need to do is post-docking analysis. Post-docking minimization, first of all. And what we do in post-docking minimization is to still do exactly what we did for only the protein and the ligand. Now, we start by, this is our protein and ligand here together. We go to you can actually click on your ligand so that you can see what is happening to the ligand. You can do control click, then up key, you can see them. We are not concerned with this ligand. We are concerned with this one that is docked. I'm even going to delete this if care is not taken. So you go to tools, you go to surface structure editing rather, you go to minimize structure. Once again, like we did before. Now we are going to minimize, this is the protein, the first one. Oh, no, the protein, because I first imported the ligand before the protein. This is the protein here. And we are interested in this ligand. That is the first docked ligand. All 10 of them are listed here. You can see it, but we are only interested in the first one. So we are going to take our protein. We are not going to take the prepared ligand. We are taking the docked ligand and the protein. We do exactly the same steps we did before. We go to minimize. Everything should be the same. It's still the two of them. Okay. Now here it says consider each model in isolation from others. No, we want to minimize both of them as together. So we unclick this. Okay. And then the last pop-up. Everything is still the same. Okay. And it goes through the same processes once again. But don't worry, this is the last time. When it's done now, we are going to see how we can present our result. We've already seen the results, so you should already have taken note that your binding affinity for this thing, the highest binding affinity was -7.3. Like I said, if I am interested in the conformation, I can already look at the conformation, binding conformation of the standard drug to this protein, and then I look at all of them, the 10 of them, and see which one has the same conformation with the one, and I pick that one. It depends on what you're interested in. For now, what we are doing is just picking the one with the highest binding affinity, which is the first one, irrespective of the conformation it has, and we minimize it together with the protein. And then we are going to see how we are going to present the information after it's finished running. So this is a time to brew a cup of coffee if you want to, or to ask some questions if you want.
Please, I want to ask a question.
Yeah.
Well done, sir. You're really doing a lot. Thank you.
You don't need to call me sir. My name is Darlington.
Okay, well done, Darlington. You're doing a lot. Thank you. I already, I told you I was having issue with the camera, so I've installed it now, but I've gotten to the place where I had to minimize. So I just want you to just refresh me that I've deleted solvent. Am I also to add hydrogen? Like, I don't know what else to do on that page.
So you delete solvent, delete non-complex ions, add hydrogen, add charges, change all of those non-standard methionine, UMP, CMP, and all of them, change them to the standard form, all of those things, click it. That's it, then you minimize.
Okay, thank you.
Yeah, for the other pop-ups, you can still tell me, and if I can remember what is there.
Okay, thank you.
Darlington, I have a question.
Yeah.
Yeah, thank you so much for breaking down this thing so much. My question is, in trying to analyze some phyto compounds, which of the compounds is ligands? Like, would the HPLC serve best or GCMS compounds?
Please, let me get your question again. Can you repeat it?
Which compounds are best used for docking from plants mostly? Is it the ones gotten from HPLC or the ones from GCMS? Or both can be used?
I'm still trying to understand the question. Compounds gotten from HPLC. Compounds are not gotten from HPLC. HPLC is just used to either quantify compounds or to separate compounds. If you want to discover new compounds, it's either you are using X-ray crystallography, mass spectrometry, NMR, or there's one more. So anyhow you identified any compound doesn't make any difference. I mean, all you know is that if a compound has been proven to have a particular activity that you're interested in, you can always try to look at that compound and see how well it interacts with your protein of interest. So we do not talk anything about the sources of those, the sources from the plant, but the technique used in identifying or characterizing those compounds, we did not really talk about it. I don't think it's of much interest to us, but these techniques could include not HPLC. HPLC is not used to, what can be used to characterize or to identify a compound. Well, identify
I'm not
on what
talking about identifying. You know, you can use HPLC to know certain compounds that are found in a particular plant you want to analyze, you want to use to do certain study. So I wanted to know, and you could also use GCMS, you understand?
Okay, let me explain HPLC a little. HPLC is that you already know what the compound is. The compound has been identified, but you want to see the compound in your extract. You want to quantify it in your extract. So you put a standard of the compound. So you can't use it to identify. If you say identify, it depends on what you mean by identify. If you are identifying a compound for the first time, no. If you want to identify it in your compound, like you put a standard of a compound in your HPLC, you see where the peak falls, and then you put your sample, and whichever peak you see there at that detection time, you know it is that compound. It's not really identification of compounds per se. It is identified because you're identifying your plant, in your extract. It's not that you are finding out a compound. If you want to find a new compound.
No, it has nothing to do with the docking. We're not trying to find out, you understand? It's in the plant. We want to know. You're not getting my question.
Yeah, I think I didn't get your question.
Like to discover new compound in that plant.
Okay.
You've heard that this particular plant is maybe good for treating malaria. And you now want to do the in silico analysis using the compounds that are in that plant that are already known. You understand? So if you are now going to get it, whether you could also use GCMS to find out the same thing. So that's why I'm asking whether you could use both processes to get at the compound you want to dock, or is it a particular process that we use?
I think you still do not really understand what I'm saying. I think somebody.
Can I come into that question, please?
Yeah. Okay.
Okay. Concerning the compound you're looking for, the issue is not whether you're using HPLC or GCMX. Your choice of using HPLC or GCMX, I think it actually depends on other factors apart from the compound. Identifying the compounds in your plant is not even it is actually up to the detector that is attached. In this case, when you said GCMX, it is MS. And then if you're saying HPLC, it's either HPLC hyphenated to UV or HPLC also hyphenated to MS. That's actually helps you to identify the compound. PC and HPLC does is that it can help you separate. Then the mass spec or the UV can help you to identify. But then, depending on the kind of plant, you can now choose whether to use GCMX or HPLC. I know basically that most of them use GC if the plant is actually an oily plant. Most of the times, it's usually preferred to use GC if the plant is oily compared to HPLC. So that shouldn't affect this whatever docking you want to. You can use it to identify the compounds in the plant, but it's not actually the HPLC that does GC. What they do is it is now the detector, which is either your UV that identifies the kind of compound that are already presented. I don't know if you're clear, sir.
Yes, I do now.
I think I still want to reinforce something there. I think we sometimes we misunderstand HPLC because I work with HPLC. I do my master's with HPLC. Okay, HPLC, like Israel is trying to explain, when you are attaching HPLC to LCMS to MS to mass spec, it's no longer HPLC. You can call it ultra HPLC or you can call it LCMS. That is what you can use to identify compounds, new compounds. You cannot be subjecting your sample to HPLC in order to identify compounds in the, no, it doesn't happen. It's not even the purpose. HPLC is just a separation tool or a quantification tool. Actually, I used HPLC for separation. I did what we call, because we have preparative HPLC and we have analytical HPLC. I did preparative HPLC and I understand how it works. So if you have a compound and you already know the compound in your plant and you want to quantify it or you want to separate it, then you can use HPLC. If you want to find out what are the compounds in this plant, I don't know them, HPLC cannot do anything for you. It's either you are attaching the LC to MS or GCMS or X-ray crystallography or NMR. These are the things you can use. Never HPLC. It never does anything for you there.
Okay. Thank you.
You're welcome. So I think the docking is done. So let's go to the final part of our work. When it is done, what you do is to save as PDB. So as PDB, yeah. So the both of them together, we go to save PDB. Remember, we are going to save our protein and the first system. Yeah, remember to go to this place because normally it should be as multiple files, but we want to save it as a single file. Okay, and then we can name it as our doct protein or doct cause depends ligand.pdb. You can save. Then we can save only the ligand, only this our ligand that is docked to this protein. We go again, we go to save PDB, we pick only our docked ligand as a single file. So it is saved in relation to change the reason why this thing is happening is because I imported, remember when I finished preparing the compound, the ligand, I now imported the protein. Normally I should have removed it, import the protein first, then import the ligand. So whenever I'm saying save in relation to the ligand, no, I'm saving it in relation to the protein. So I save. Oh no, because I did not change the name. It's save. I'm saving the ligand in relation to the protein, and the name is going to be docked ligand.pdb. I save it. So I am not going to go into the 3D presentation. It's going to take a lot of time and it's not interesting. Let me just touch on it very small. Then I'll go to 2D, which is actually much more interesting, much better way to present your data. For the 3D presentation, let me delete this atom because it's not doing.
Yes, let Mr. Simon say current affairs.
So
yeah, that's what you should read.
Zichat Kuyet, please can you mute yourself?
So there are different ways of presenting this. It's not gonna agree. You can present, if you are presenting as a publication. Now I'm going, please, let's not go into this because it's gonna take all the time in the world and I'm feeling very sleepy. What I'm going to do is let's go into 2D. Maybe the next time we do the blind docking, then we do this 3D presentation of this one. It's not something interesting to me because as somebody who is into data analysis, I understand that it's not about how beautiful your work is, but about about the information that you can get from it, because here, even if we present it, you cannot see the kind of bond that is present and all those. So let's go directly to presenting our work in 2D, which is very much informative. And that is going to take us to the next tool we are going to work with, which is our Discovery Studio. So this is the Discovery Studio. This, it might be here when you start it for the first time, so you need to take it to ligand, receptor ligand interactions. There are so many things you can do with this tool, but for now we are just going to do receptor ligand interactions. So I first of all import my protein. The protein I'm going to import is the prepared protein. I'm going to import my prepared, which is immediately after preparing the course, the one I did before I even started docking and all those things, my prepared course. Then I need to import my ligand, and the ligand I need to import is the docked ligand. That's one that we selected, that one that has minus 7.3. That's the docked one. That's the one I saved last. I need to import my docked ligand. So I come here and copy it just by right-clicking, copy, and go to the protein and paste it. So now you can see it is here with the protein. So what I can do is to click on this button here, and then I want to define my protein and I want to define my ligand. So for my protein, this is my protein. You know, click on here, it has accepted. You click on your ligand, this is my docked ligand, then you click on define ligand. It has also accepted it as my define ligand. So we can now see how the both of them has interacted. If you look at that, let me decrease this. If you look, how do I do this? If you go back to this, you will see that this is exactly how they interacted. It's wrong. This interaction here is exactly what we have here, because that's it here. So you want to save it, you can turn it to a white background if you want. Background white. If you want to still save it in 3D format, but for me, that's not really something I want to do. Okay, that's not really what I wanted to do better is to go to 2D diagrams and click on 2D diagrams. And it has shown me how this interaction happened in 2D. It did not just show me how it happened in 2D, it's showing me the amino acids that are in this place that is bound and the kind of bond in those amino acids. For instance, there are only 2 bonds for this particular one. You only have the van der Waals bonds and the acyl bonds. So it forms acyl bonds with this, and you see the particular groups of this is the ligand binding with amino acids in the protein. And these ones did not really show the interactions where it's binding to, but it binds with van der Waals. If you do so many different you see so many different interactions, the conformation of this will change. If you bring another, because this we saved only the first one, if you bring all the others, you will see how they interacted with the protein, which is much more informative than doing the 3D. We can still make the 3D to be informative to see the amino acids in it. Like I said, we discussed that some other time. We still see the amino acids, we see the interaction with the amino acids, but it is not going to tell us what kind of bond they are binding. It's just going to show us the amino acids and the active site and the sites where we bind it, because we don't know if it's the active site, and then shows the bonds between them, but we cannot get this information of what kind of bond is this. For me, I think that this is a better way to represent our data. The next time so remember you have already seen what is the free energy of binding from the first result that showed. You already copied that down. Now you can also present it here in 2D format. If you want in 3D format, which you can edit and show the amino acids and all those things, you can also do it. That's the way to present your results. So this is what I did in my thesis. It's not really supposed to be part of my thesis. I added it as an extra thing to do in my thesis, and it was informative. Personally, I liked it. So today we've talked about how to identify our targets. We've talked about how to download these targets in appropriate formats from different repositories. We've talked about how to prepare our targets, our protein and ligand. We talked about how to dock them, how to do post-docking modifications. We talked about how to do presentation in 2D format. We did not discuss in 3D format because of time. I know you are tired and I'm tired. I've seen that so many people have already left because it's getting too lengthy. So I don't know if you have a question at this point that you want to ask. You are free to ask, please. I don't even know if the meeting is still on.
Yes, the meeting is still on.
Oh, okay.
Okay.
Sorry, Darlington.
Let me stop sharing my screen. So,
can I speak?
Yeah, sure.
Thank you very much for the presentation and everything. It's not really a question. I just want to plead because I was not there when you were talking about the downloading of the camera. I don't know if Vina is also a software, or I don't know if the information can be shared again on the WhatsApp, because I joined just minutes before I started the Zoom class. So I don't know if you can just throw a highlight, just download those stores, how to save them. I was confused when you were talking on how to save them on everything, because I was not there when you gave the instructions of the download the first time. So I don't know if it's possible, you can share it on WhatsApp or you can talk about it here.
So the group you joined, just click on the group descriptions and you see all the information that you want.
Okay.
The group description, I have some links there, the link for installation of the software, the link for the meeting link, and the link to join the group. I think, no, it's already the group, so there's no need to add the link to join the group. So you can see some links there in the group and you can use them. I think it will give you everything that you want.
All right, thank you.
Thank you.
Okay, thank you very much, Mr. Darlington. Thank you for the time, and it is very, very insightful. But I think the most important thing is that number one, for someone like me, the network is really disturbing. Because now I was logging off, coming back, logging off, coming back, so I couldn't follow the whole thing. So, but I think that you can just help us with this recording, then give us like a week or 2 weeks interval to sit down with the videos and practice all over again. So by our next class, which will be the second class, everybody will be on board like that. I think that's very, very important.
Yeah, sure. I already said the meeting is being recorded and I'm going to release the recording as soon as it's available. I'm going to upload it maybe to a drive and then send the link to the drive to the group. Anybody can access it whenever you. As for saying you would come back, you don't really need to come back. I mean, if you learn how to use it for your work and you want to work with it, you're always free to do that. You don't really need to report back like as if we that you have a test or examination for it. No, if it's something you find useful, you can use it to do your work. Yeah.
Okay,
Yes.
can I ask a quick one, sir?
Yeah, you can ask a quick one if you delete the SAR, but if the SAR is going to be there, then.
Okay, Darlington, calm down. All right, so you talked about 2 things from the beginning. That's the druggability of the receptor and also the drug likeness of the ligand. Okay, and you helped us to understand how to know if the receptor is druggable based on the fact that, for instance, the COX we worked with, it has actually been a receptor for most drugs. So the likelihood of knowing that it's druggable is there. But you didn't really throw light on how to know if a ligand has the drug likeness. I think I know something about the Lipinski rule of 5 and all that. Is there a way we can identify those stuff with the software? Well, the control light on that.
Yeah, so for instance, before you start picking a ligand, you should already have seen that this ligand has been used in some works. For instance, it has already been identified that it has social properties. It's not something you can just see any compound and start using. It's either something you are working on already, you've done your LCMS and you identified some compounds in your plant and you want to see, can it really interact with this? You can, or you have already gone online and you have seen that people have identified that this plant, I'm using plant now because it's the example, this plant has social properties and these are the compounds identified in the plant. Even if there are compounds identified in the plant, it doesn't mean that those compounds must have the properties that you're looking for. So you can take the compounds and read about them. If you go to PDB, so not PDB, PubChem, or you go to any other of these things, they give you some information about the compound, some of the activities, and they mention that it has an anti-inflammatory activity, then you know that, okay, it's already known that it has such activity. So you can read about the compound. And I think that one is even easier to find out than even to find out if a protein is druggable, because if a protein has not been used, has not been targeted by a drug, like those potential druggable proteins, how to find out whether they are druggable is actually very difficult. But if it's for the compounds, somebody must have worked on them and find out that these compounds have these properties before you start using it for a document. Unless you are doing your own research. For instance, I was doing my own research and I did LCMS. I did HPLC first of all to isolate some compounds, and then I did LCMS to identify what these compounds are. And then I wanted to see if these compounds have activity on a particular protein I was working with. And then, so because some of these compounds are not even non-compounds. I'm the one that identified it for the very first time. So I just tested everything. And some of them did not give me what I want. Some of them gave me what I want. So in that case, you might not even know whether it is drug-like or you are testing it. But if it is just a work that is not your own research, you want to test, you should know the properties of those ligands before you start so that you don't waste your time.
All right, sir. Thank you so much. So then one last thing. Probably maybe you could just give us an idea of how to make inference from whatever we get from this.
Thank you very much. So I was going to come to that, and sorry I didn't start with it earlier. So this, I believe in collaboration. It's very, very, very important. If your target is to do some talking studies that you can publish to add to knowledge, because I believe, like I said when I started, that if you are trying to get scholarships, you want a professor to hire you in their lab, you should show them that you can do research so they can invest in you. So this is something you can leverage to, and I don't believe in working in isolation. You should look for like-minded people in this group. This is one of the reasons why the WhatsApp group is created, where you can find on other people who have interest in working in something. Let's assume you want to test 200 compounds. When you people are together, you do it, these people do this, these other ones do this. It's much, much easier, and you can come up with something publishable. Another thing you can do is to link with people who are authority. I learned this from my supervisor, because when I talked about molecular docking, I brought the idea that I want to work on molecular docking on these compounds, and she was like, well, if you can do it, fine and good. And then she told me that if you're doing it, why not go online and find people that are already authorities in molecular docking studies? And then we can invite one of them to do one or two things in the work, and then so that their name will also be in the publication. It helps the publication to move, to go higher. So I learned that as well. So if you are partnering with people in the group, you can also look out for people who are in the field. When you design the study you want to do, you can even give it to them and ask them to look at if your design is okay. Because it will not be nice that you finish all the work and then you send them and say, I want to add your name to it, so that they be part of it. When you design your study, you give them, they confirm that, okay, the method you have selected is okay, this one is okay, this one is okay. And then you work. After working, you send to them, they confirm it's okay. You publish together, their names are there. So the work has some, weight because an authority in the field is already in the work. I am not an authority in the field, for sure. I am just learning this. So you can look for people who are authority in the field and has, so I encourage people to collaborate in the group, find people who are willing to work on what you are working on. Personally, not me, because I am extremely busy now. But you can find other people in the group. I really encourage people in the group to merge up, 5, 10 people, work on the project, possibly get a very nice project and publish it, and it's a very good thing.
Darlington, can I ask I want to ask, if you're trying to kind of compare you're looking for a new drug from a plant, right? But a standard drug, and you get to dock the standard drug to the protein as well as the new compound you're using. What will you use to tell which of them is better? Is it the negative energy we got, like the affinity, that you tell if this one, like you let's say you just docked a compound now from a plant, right? And then you're trying to compare it to a standard drug you have docked with the same protein. So what are you going to use now to know if the drug being used has better binding and is better, it serves as a better drug than the plant which you're using? Is it the negative, like the one we just
like the free energy, the energy of binding, the binding energy is the most important thing. But like I said, you might want to do a side-directed docking to be sure that this ligand is binding in the same site with your drug, so that you know that. You can also look at the structures, because one thing I do is also to bring the structure of the standard drug. When you look at the standard drugs, you can see that some of them has peculiar structure that makes them to work in the same way. If all of them are cause inhibitors, you see that they have all of their structures, they have something in their structure that makes them to really inhibit cause. So you can also look at your compounds and look at and see if it has such a structure in it, so that you see that okay, yeah, it has something similar to them and it's even binding better. So the binding affinity is very, very important. If you don't look at the binding affinity, well, it might even be binding somewhere else, like allosteric enzymes. It might be binding somewhere else in the enzyme that can still inhibit the enzyme from working, but it's not going to be a competitive inhibitor of the drugs that are, not the drugs that are used, the molecule which the enzyme works on. Because now I should have explained this. The standard drugs, what kind of inhibition are they exhibiting? That's one thing you ask yourself. Are they competitive inhibitors of the substrate or the enzyme? If it is yes, then you also try to look if this your compound is possibly a competitive inhibitor as well. Does it have a similar structure to, because you will find out that the inhibitors that are used as drugs are usually structural analogs of the substrate. So does your own compound have a similar feature? If not, it could be binding. It could be binding, because for now you don't even know if it's going to elicit a physiological response. If it's going to really modify the protein, all you know is that it binds higher than. But if you do site-directed docking and you see for sure that it's binding in the same site as the drugs, even if it doesn't have a similar structure, it's binding in that same site. As much as it has a firm binding on that site, it means that it is going to compete with the substrate. We can't be very sure of this. This is the reasoning. And that is why this is just like a preliminary screening. Later on, in drug development, if this is really the field you are interested in, I'm going to recommend the course to you. If you finish this course, you will really understand how these things work very well. The name of the course is The Science of Medicines.
Science of Medicine.
The Science of Medicines is taught by Monash University, and the course is on FutureLearn. It is one of the best online courses I've ever done in my life. It explains the process of drug discovery, both the chemistries, the therapeutics, the pharmacology of the drugs. It explains everything. Like, you would really get to understand really how drug formulation comes about and all those things.
Okay. Then so The site, you said something like site-dependent binding or what? Site-specific binding. I think when you're talking of trying to know.
Yeah. Site targeted binding, like to target the active site of the enzyme. So the difference between site targeted binding is that unlike this one that we are just using a grid volume where we now interact, like we saw 10 different bindings in different conformations and the protein. This one is that we define exactly what is the active site of our protein. And so when we are binding, it's going to bind directly in the active site of the protein. We are going to use a different software altogether to do that. I don't think you can do it with this software we use now. It's also a different course I did to learn that. So we can do that some other time. Frankly, I don't know when because my schedule now is tight, but we can find time to do it for sure.
All right. Thank you very much. Thank you.
You're welcome. So I don't know if there are other questions in the absence of other questions because this is already, morning. Yeah, it's already morning. So
morning and happy Sunday.
You see. So any further questions before we call it a day?
I think if any other person has a question, we can address it on the WhatsApp group.
Okay, that's very good. Like I said already, I will not be because I have a full day tomorrow and I need to sleep this night.
Today.
My day starts today. So from Monday I can start responding. And from Monday, I don't also know how it is. I started a new internship and I don't know how busy I will be. But the truth is, whenever I can, I will just come in and answer any question. The ones I know the answer, you can see for sure that I am not an authority in this field. It is just something I learned. I think that others can benefit from. So after today, you might know this topic better than I do. So if somebody asks a question, please try to answer. If you know it, I might even be the ones asking questions because some things are not clear to me, for sure. So please, if I ask questions, don't tell me that I am the person that taught you. Please, if you know it, answer me. Thank you very much. Thank you for the paper. I saw Latifa, was it Latifa that asked the question? I think Latifa asked the question sometime. I don't know if, yeah, surely I did not answer your question because surely I did not answer your question, but I think I have hinted on what I know about it. And for every other person, thank you very much for joining. I really appreciate it.
All right, guys.
Yes, back to you.
So much, Darlington. Please, guys, help me thank Darlington for such an amazing 3 hours of awesomeness. Thank you for impacting this knowledge in us. We pray that the good Lord will bless you and perfect all that concerns you. And then we also pray that this thing that you started will lead to several publications and, of course, several people who get as many scholarships as possible in different parts of the world. Thank you so much. So guys, we're going to try to upload this video right now. It might take some time because it's a very large one, but once it's ready, we would send it to the group so that everybody can partake of it. And of course, if you've gotten this whole idea and you are interested in collaborations, the group is still open. Like Darlington said, a lot of people might be interested in collaborating with you and having publications. So Darlington, thank you once again. Thank you. We really appreciate the time that you've spent to give us this knowledge, and we do not take it for granted. Thank you so much. God bless you, sir. And everyone that partook of this section till now. I know when we started, we were like 60 now. We are almost 20 remaining. So thank you so much, everyone. And do have a wonderful night rest. Thank you, guys. Bye.
Bye.
Nancy, do you have any last words you want to tell us before we go? Yeah, run away.